All right. Hello, everybody, and welcome to another Black In panel. So today, our Black In Marine Science panel is going to be hosted by me, Symone Barkley, and Carlee Jackson. The Black in Marine Science effort and week was started really for Black marine scientists and allies to organize and coordinate an online event to engage all Black marine scientists and our supporters. This is something that we're really excited about doing, and the National Aquarium is really honored to be a part of this effort. And so to get started, just to give a little introduction about myself, I am one of the managers of education at the National Aquarium. My history really includes me studying sand tiger sharks and sandbar sharks in the Delaware Bay, looking at their interactions with anglers. And I'm really happy, right, to be able to talk to these other Black marine scientists who are doing really outstanding work. The panel that we're doing today is plants versus animals, because the ocean has so many different species, and we always love a good panel here at National Aquarium. And today I have Carlee moderating with me. So Carlee, would you introduce yourself for everyone?
Yes, so my name is Carlee Jackson. I am one of the organizers for this wonderful Black in Marine Science Week. A little bit about myself: I just graduated from Nova Southeastern University with my master's in marine science, and there I did shark research. I looked at the effects of provisioning tourism on nurse sharks in a little island in Belize. And I also work with marine sea turtles. I've been working with them for a couple of years. And I am really excited to get started with this panel and learn more about all these wonderful Black marine scientists.
Okay, and with that, we are going to get started and have each of our panelists introduce themselves. And we will start with Katherine.
Hi, my name is Katherine Hannibal. I'm a current master's student at the California State University, Northridge. Generally, I'm really interested in how humans affect coastal ecosystems. And I currently study how marine invasive algae affects invertebrate life histories. So I study mussels, oysters. I get really excited about little tiny things.
Hello, everybody. My name is Colin Howe. I previously earned my master's degree in marine and environmental sciences at the University of the Virgin Islands in St. Thomas. And I have been able to continue my education. I'm now currently pursuing a PhD in biology at Penn State University in Dr. Monica Media's lab, focusing on coral microbiomes. So understanding how these microbiomes influence coral physiology. I really love corals. That is definitely my thing. I've been able to study corals around the Caribbean, and I love acropora corals. You can see the example in the back is a hybrid taxa called Acropora prolifera. So that's just a little bit about me and my interests.
All right. Hi, everyone. My name is Danielle McHaskell, and I just recently graduated from Cal Poly Pomona with my master's degree, and I'm in a second-year PhD student at the Scripps Institution of Oceanography at University of California, San Diego. So as far as my interest, I love seaweed. So I'm really focusing on how invasive seaweed species might impact the biodiversity of native ecosystems, particularly here in San Diego and temperate ecosystems. So I'm very much involved in kelp forest ecology and then intertidal ecology as well. Recently, I'm starting a new project now in the PhD program, focusing on how an invasive species, Undaria pinnatifida, might affect the area here in San Diego. And it's been a real joy. I'm deep diving in the literature right now. I'm having a great time.
Hi everyone, my name is Shalanda Grier. I'm a fourth year PhD candidate at UCLA, and my research interests just circle around abiotic drivers in tropical systems such as coral reef systems, but specifically looking at macroalgae and understanding how macroalgae respond to changes in abiotic drivers, and also how resistant they may be to these changes in coral reef ecosystems.
Hi everyone, I'm Sennai Hastes. I'm a research assistant professor at the University of the Virgin Islands in biological oceanography. I completed my PhD at the University of South Florida, where I studied habitat modeling for Atlantic bluefin tuna. And now most of my research focuses on understanding oceanographic influences and environmental influences on the dispersal patterns and abundance and distribution of early life history layers or stages of fish. So the larvae, the small juvenile baby fish. And so we use that to help inform the decision support system for the management of important coral reef fish here in the Virgin Islands and the rest of the US Caribbean.
Awesome. Sorry, y'all. I had to take a second to get off of mute. So awesome. We're so happy to have all of you with us. And now we are going to get started with our questions. And Carlee's up first.
Gotta unmute myself. Typical Zoom. All right. So the first question for you guys is, how did you get interested in your research organism? So what interested you about your research organism?
So for me, I study mussels and how this invasive algae, Siphonia japonica, really affects their life histories of larval mussels. And I think what really got me interested in it was in my undergrad, I was helping out with this really long-term survey that they were doing of this one bay that had been completely just decimated by a lot of nutrients. The river system was completely out of whack and we didn't have a lot of policy surrounding it. So we had a lot of blooms of a lot of different algae. And one of the days we were kind of just walking around in the middle of the winter looking for different algal species, and I saw this really fluffy red. And I mean, that's not usual for middle of winter. And so I got really excited about what it was and did some research and found that it was Siphonia japonica, and that it had invaded the space that we were in. And when I got to my graduate studies, it was kind of in the back of my head of like, what's going on with this algae? Has anyone really studied this yet? And there were a few papers out about it. So when I was working with my graduate advisor, he was kind of talking about his research and how he studies a lot of mussels and how we have an NSF grant to study mussels. So I was kind of trying to figure out where my interests fit in with mussel beds. And I didn't really think mussels were something I wanted to study. They're just kind of there in my mind. But as I started really diving into mussel beds and their functions and how important they are to the intertidal, I remembered this algae that had been popping up and really wanted to look at the differences and how that algae is affecting mussels overall. And no one had studied it yet. So I thought, you know what, I'm gonna study it. And so I did. And that's how I got interested in my subject.
Great story, Katherine. How I got interested, I've started studying a bunch of different things. Like my first species ever I looked at was working with invertebrates like lobsters and crabs. So then I started learning a little bit about lionfish. But I think what really got me hooked was an experience I did. I was a TA at a study abroad located in Bonaire, which is in the Dutch Antilles, next to Aruba and Curacao. And part of my responsibilities was to TA college classes and help and assist in teaching that and teaching students coral ID. And then I also participated in a long-term coral monitoring program that basically quantified, percentage of coral, how much coral cover was out along the really beautiful, diverse habitat in Bonaire. And so I was constantly just kind of relearning all of my different and reinforcing the different coral species. And then I was able to luckily earn my first publication studying how divers impact coral reefs, the structural complexity for coral reefs. And that's kind of what got me hooked in. I was like, okay, yep, corals are it. And once I continued my education and started working in the US Virgin Islands, we learned a lot about Acropora corals. And Acropora are a group of corals. There's actually 2 species and a hybrid taxa that is really popular, is really common in some, or it's not common actually, but it's critically important, and it's also endangered in the Caribbean. And so I really was fascinated about this, and learning about some of the ecosystem services that these corals provide is really important, and it has an application to benefiting, not only the ecosystem, but ourselves, the communities in the Caribbean. So that's what really got me interested in these particular species.
Thank you so much for sharing, Colin. So for me, actually, although I know I'm here as a plant seaweed person, I actually was on the other side and really loved animals. So during a community college field trip to the Intertidal zone, it was my first time ever being in the Intertidal zone. And there was this beautiful little sea hare, and then I picked it up and just fell in love with it. And so when I was an undergrad and transferred to a 4-year, I knew I wanted to work with sea hares. I knew nothing about research, but I was like, these are really cute animals. I like learning about feeding dynamics. So yeah, that's gonna be my project, I guess. And waltzed into my PI's lab and said, I don't know what research is, but let's do this. And luckily that undergrad research, he said yes, that undergrad research transitioned into my master's thesis. And so for that, I was looking at how this giant sea hare called the black sea hare can be as big as 30 pounds. So really cool sea slug. I know they're humongous. So it was looking at how this sea hare's feeding dynamics might change depending on what species of brown algae you feed to it. And two of the brown algae species that I was using was Sargassum horneri and Undaria pinnatifida. So both of these seaweeds are non-native seaweeds. They're invasive seaweeds from other regions. And it was just really interesting that although these two seaweeds do have different morphologies, one being more weedy, the other one being a kelp, it was interesting that the black sea hares, although very voracious, they eat a lot. They did not really like Sargassum utica or Sargassum horneri. Didn't eat that one as much, but loved Undaria pinnatifida. And it was interesting because Undaria isn't really studied very much, and we don't know very much about it in the US. There's been no studies done in the US looking at how it might impact the Native community. And there's only been about 5 studies done in the US, particularly in California, on Undaria. So it seemed like this really cool like niche that I can fit into. And so I was really interested in working with Dr. Jennifer Smith at UCSD. And so I just started talking about Undaria, an invasive seaweed species. And I just realized like, yeah, this is my place where I belong. I just love learning about how seaweeds, which you think are these very boring, like just standstill organisms, actually compete with each other for resources. And I just found it very fascinating. So yeah, this is where I am for the next 40 years.
Thank you for everyone's story. It's so interesting to see how everyone kind of got into their groove. I think how I became interested, it was merely by accident. I won't say that like I started my undergrad interested in marine biology at all. It would just happen to be an internship that I took part in called the Diversity Project, where I got my first experience like snorkeling, going and just seeing a coral reef ecosystem for myself, something I've never seen outside of a textbook. And similar to Danielle, just like the thought process of people just thinking, algae are just these random, boring organisms in the ocean. Prior to this experience is something that I would have been scared of, like just not being able to see like in a more like temperate region, not being able to see what it is like, oh, this is algae, but actually participating in that internship, swimming around and observing the different macroalgae and just learning that they have really important functions in the system that they're in. And knowing that, and sometimes in tropical ecology, tropical coral reef ecology, macroalgae can sometimes get a bad rep, but they're not always bad. So my first species that I worked with was Halimina apuntia, and it's this really pretty like green branched algae, macroalgae, and just doing a project, learning more about what that species provided to its environment and how it responded to just different abiotic disturbances really got me interested in learning more about macroalgae and like kind of telling the story of macroalgae in a different light. So yeah.
So thanks, Shalanda. That really warms my heart to hear. This is one of those great examples of how the breadth of the work that Black marine scientists do is so wide, but the network is so small, because I was one of the co-principal investigators on the project where Shalanda went to do some of that research. So it's really great to see how far she's come and how that influenced the work that she's doing now. Personally, for me, I think I would say that my pathway into marine sciences was definitely not a straight line. You know, when I went to undergraduate at UNC, I had no idea that oceanography was even a field that I could go into. And so, you know, I had a lot of really strong mentoring programs that I attended that kind of helped guide me in the direction of where I ended up, which is now being a fisheries ecologist and fisheries oceanographer. And through graduate school, I always knew that I was interested in understanding how abiotic, meaning non-biological things, affected organisms. And one really great way of looking at that was in zooplankton and ichthyoplankton, which these are organisms that live in our oceans or in our lakes and rivers that aren't able really to swim or move with a lot of power on their own. We often separate those into, you know, phytoplankton, which are plants. They make their own food due to sunlight, and organisms that need to feed, meaning zooplankton or ichthyoplankton, which are fish organisms that are in their larval stages. And so I got into that because I was using, in my master's program, I was trying to understand how an imaging sensor, basically a camera that we towed in the water, could be used to identify organisms. And I just found the diversity and the abundance of organisms that we don't generally see with the naked eye in the water column in our oceans to be just really amazing and full of interesting questions that could be asked about them. And so for me, that was really the driving factor, learning the meristics and morphometrics, which are basically the attributes about these organisms that make them different from one another. Learning those and understanding how to identify different species really pushed me in the direction of the work that I do now. And I grew up in the Virgin Islands. I've always been like an avid water person. I love fishing recreationally. And so figuring out research that allowed me to use those interests in research questions about the diversity of organisms that live in the ocean at that small scale, but could inform fisheries management was really important to me. And so I decided to work towards what we call applied research, or research that solves problems directed at management that affects humans. And so for me, looking at the early life stages of commercially important reef fish was a very good way to do that. And so that's how I got into my work now on large body parrotfish, the NASA grouper, and the mutton snapper, which are all fish species in the US Virgin Islands that have been fished down to very low levels. And so understanding how oceanographic and environmental conditions affect their dispersal and affect their populations is the type of work that I'm doing now.
Thank you so much, everyone, for all your contributions. This is really amazing to hear, you know, your origin stories and fun fact. I always have to give like a little fun fact. Fun fact, my very first internship was at the Institute of Marine and Environmental Technology, which is next door to National Aquarium. And I studied algae. I studied Nanochloropsis oceanica, actually. And so I didn't continue studying algae, but look at that. There's a little connection. So the next question that we have is for each of you to tell us 3 fun facts about your research or your research organism. I'm really excited to hear some of these.
Ooh, fun facts. Okay. One fun fact about my algae is that it's both eurythermal and euryhaline, so it can survive in a really vast amount of either temperature or salinity. So it can be in like brackish waters, almost completely fresh water, to like really, really salty waters, and then kind of warm water to very cold, like Baltic water. So it was really interesting to see kind of how fierce of a competitor Daisy-Siphonia is. I like to say that it's going to take over the world. I'm not rooting for it to take over the world, but it would definitely be interesting for my own research. And I think that with mussels coming into kind of how Daisy-Siphonia is set up, it's a really important interaction. So mussels have this, my second fun fact, mussels have a primary and secondary settlement phase. So primary is like its first little larval phase, and that's what I study, and they settle within Daisosiphonia. And then secondary phase is when it settles within the big mussel beds. And mussels within a mussel bed are kind of thought of as this foundation species, which is my third fun fact, because they really just, without them, the entire ecosystem is changed. They're really important for kind of being that solid foundation. And a lot of different species settle within their interstitial spaces, so the spaces in between their threads. And I think that is so incredibly cool that they really have a large part and large hand within how our ecosystems are set up on our coastlines.
You know, I'm going to follow Catherine's lead with the foundational species here. Again, I'm going to reference the acropora corals. Corals in general are considered a foundational species, right? Their presence influences the ability for other organisms to live and thrive in this environment. And two species, Acropora cervicornis and Acropora palmata, have dominated coral reef communities throughout the Caribbean for millions of years. Fun fact number one. They used to form these large, massive thickets with a lot of three-dimensional structure, right? And that supports a host of organisms, not just fish, but other invertebrates, polychaete worms, and that attracts other big fish, which we like to eat, right? So there's a really nice foundational species there. Another thing that's kind of unique with Acropora taxa is that they have relatively quick growth rates, right? When we think about corals, generally corals grow relatively slowly. If you can imagine these big bouldering corals, or there's some brain corals that get pretty big, right? Those corals only grow, a few centimeters a year. Well, both of the parental species and including the hybrid taxa, Acropora prolifera, have really fast growth rates, anywhere from 5 to 8 and sometimes 10 centimeters in a year. And so it's really kind of remarkable to study a group of corals that can do things like that. And that's interesting fact number two. And throughout the Caribbean, unfortunately, with these two particular species, we've seen major declines. And I mean, when I say major, like upwards of 95% population decline of both of these species throughout the entire Caribbean. And so we're losing these kinds of ecological services that these corals provide. But what is also interesting, and the third interesting fact, is that there is a new effort surrounding something called coral restoration. Now, this primarily focuses on two of the Acropora species, but they're incorporating these bouldering corals as well. But they've identified to capitalize on this rapid growth rate to expedite coral growth in the wild in an attempt to reestablish these corals back into targeted coral reef populations, right? So you go out to a coral reef site, you identify if this is going to be an ideal area for restoration. You have these corals growing in these structures are called coral nurseries that promote coral growth. And then you can reattach these corals or a lot of different methodologies back into the reef. And so kind of what Sennai was mentioning earlier about this applied science, our understanding of how corals respond and how they grow to try to reestablish a lot of the degraded reefs in the Caribbean.
Awesome, Colin. Thank you for dropping all that coral knowledge on us. Like, we needed that today. So for my 3 fun facts, I'm going to backtrack a little bit to my master's undergrad work and talk about the black sea hare, because I'm just kind of a sea hare geek. So I kind of dropped one fun fact that they can be as big as 30 pounds. The ones that I worked with only about 10 or 11 pounds. But another fun fact is that they lay eggs from like a little orifice that's near their mouth. So if you when I would go into the aquarium room in the morning, after they've been alone all night, I would find these little like pink spaghetti patches like in the corner of the aquarium, and then see them kind of connected to that pink spaghetti, and it was coming out of their mouth. And so that was actually their egg structures. And so they like to lay eggs in these like little crevices to kind of keep them protected. So that's one fun fact with the black sea hare, Plesiophocaria. Switching gears now back to seaweed. My second fun fact is about Undaria pinnatifida. So Undaria is actually on the International Union for the Conservation of Nature's list of the 100 most invasive species on the planet. And now it's only one of 2 seaweed species on that list, the other seaweed species being Chlorpa taxifolia, aka the killer alga. So it's really cool that Undaria is on that list. However, we don't know very much about it, as I've kind of mentioned before. My third fun fact is that for non-native species, this includes specifically non-native seaweed species or invasive seaweed species, we only have studied about 6% of those seaweed species to determine what their impact might be on the native community, which is really important for us to know because we are going to be allotting resources for management to determine how to control the seaweed species, how to limit its spread. So we really need to understand, excuse me, we have to understand how these invasive seaweeds might be interacting with the native community, although we don't really know very much about it right now. So those are my 3 little fun facts about seaweed and sea hares.
Thank you, Danielle, for sharing. I'll just continue on about seaweed. So one fun fact, well, the first fun fact that I'll talk about is that seaweeds are specifically some tropical macroalgae have what we call like structural responses. So they are able to like kind of structurally defend themselves against like abiotic disturbances or changes. So we've seen with like increased nutrients, and usually this is thought of like as an anti-herbivory response. So like if algae are bitten, like perhaps they'll have some type of historically chemical response, but now we're seeing that they also can have structural responses, but not only to herbivory, but to like changes in nutrients. And more recently, I found that Pedina boreana specifically has a structural response change to sediment and light disturbances. So something that under normal conditions where it would become more tougher, I guess, it will calcify more. So essentially it'll be more tougher when light is reduced. The opposite happens and it becomes more foliose in nature and not so tough. So that's interesting that algae have structural responses to abiotic changes. Something else that's interesting is that Halimeda opuntia, so it's like green calcifying algae. Like I said, it's like very branched, it's like small and very cute. But this algae contributes a lot to sand. So if you go into a tropical place like where you see a lot of Halimeda, you can pick it up and you can find like little bits and pieces of like dead calcified branches of Halimeda. And my last fact is that my species that I look at, Pedina boreana, is the only brown calcifying algal species. So that's really interesting, I think. Yeah, those are my facts.
Great, thanks, Shalanda. I will try to match the interest that Shalanda provided, but I'm not sure if I can do justice. But one interesting fact that I find always really neat, because it kind of identifies that these are organisms that live in a completely different world foreign to us, is that fish start out as as eggs, right? They get hatched in the open ocean, but ocean is water, right? And so in order for these eggs to survive and not completely sink down to the bottom, get covered up by sediment and die, but to drift, they have to have something that keeps them buoyant. It has to be different than the water column. And so most larval fish start out in these eggs that have what's known as an oil globule. It's a saturated fat, similar to like, fish oil that you eat in salmons. Inside their little egg is this tiny little droplet, an oil globule, that you can kind of see when you look at these eggs under a microscope, that allow them to the fish eggs to float and drift with the currents to move to their to the areas where they finally hatch and grow into larval fish. And that's tied to the next fun or interesting fact, which is that larval fish, when they're moving from their spawning areas to where they end up settling or where they become parts of the population, can be separated by distances of hundreds of kilometers, meaning that marine protected areas that have fish that spawn can be supporting the population of fish several hundreds of kilometers away, because their stage where they're growing into a swimming and motile juvenile fish can take months in some species. So, understanding the pathways that these fish take and the different types of conditions that affect them are some of the things that really interest me in my work. And then a third really interesting fact that is more germane to some of the species that I look at down here in the US Caribbean is that most of these are broadcast spawners that go back to the same place on an annual basis, meaning the adult fish go back to the same spot within a distance of 1 meter to spawn every year with all of the rest of their adult species, or the adult parts of the population, which I think is just crazy to think about an organism like a fish that doesn't have anywhere nearly as developed a brain as humans do, but can navigate in the ocean better than I can using a GPS to a spot every year to reproduce, you know? So I would say those are my 3 interesting facts.
I just have to say that I was so like intrigued while all you guys were talking. Like, I haven't really learned much outside of like the shark and sea turtle world, and just like learning about all these invertebrates and everything is just so cool. And what you guys are doing is just awesome. So, and that 30-pound sea hare, I'm gonna have to Google that because that's a little crazy. But all right, so here is you guys' next question. So what are some of the challenges and rewards that you've faced in your research?
Ooh, that's a tough one. I think some of the challenges that I faced, and it seems that we all are doing some kind of field work. Field work in general is a challenge. When I was doing my research, I do it all in the Gulf of Maine, and specifically off the coast of Boston. And hurricane season is directly when I do my research. At the peak of hurricane season, actually. So a lot of my plots have gotten lost. We've had to go and recover a lot of different equipment, but it's kind of all worth it in the end. When we're all able to get the data that we've been hoping for, that we've been planning for. It kind of makes all of the challenges of just being in the field worth it. But I think like a real challenge in just the world of marine science has really been like finding my place and my people and like the space that I fit into, like my specific niche. So it's been, I think Twitter actually has been really great at connecting me with a lot of different people, a lot of different people that, are interested in my work, that, study invasive algae, people that look like me. I never really, I understood the importance of that, but until I was really sitting down in a room with a bunch of Black marine biologists, it was like a spiritual moment. And so I think that that's one of the challenges, is really finding your place here as a Black marine biologist. But the reward is being connected to a lot of amazing people, and being able to really have a team behind you that understands what you're going through or can mentor you in different ways. Danielle was actually one of my mentors at the Western Society of Naturalists conference a few weeks ago. So it was really great, like connecting with her and understanding kind of like where I want to take my research next, what I want to do further in my career, and having that mentorship already built in. And I think another reward is just being connected and knowing that giving back is important, and going through outreach and community science is really important for building the next generation of Black marine scientists. So it's been really rewarding for me to be able to be a part of some really great organizations.
Wow, nicely said. It is a little tricky. Field work, I think, is a given for sure. I've definitely had some of my just terrible experiences overcoming, because the ocean is very unpredictable. And sometimes it's hard to, especially if you have your corals outplanted and something destroys them. But I think some of the challenges that are applicable across STEM fields or just science in general is, especially when I started out, learning how to incorporate feedback and criticism. And I think it's important sometimes for me to remember that science inherently requires that, right? Like process of science requires you to work your butt off towards some specific goal and task through your own experimental design, and that people in the field question and challenge your ability to arrive at this conclusion. But then, through this kind of deliberation and this kind of discussion, understanding how to articulate yourself in science and in language, I think, is the reward, right? This is my mom's quote, that adversity makes you stronger, right? And I think that it is inherent within science to have challenges. Challenges are always going to be coming at you. And something that Katherine mentioned earlier as well about outreach. I think as scientists, another challenge that we face is trying to disseminate science and have people understand that climate change is, something that it affects us all, mostly and specifically people of color. But then, incorporating a diverse group of individuals and ideas from multiple backgrounds to come out and, kind of tackle these solutions and then come up with effective ways to communicate these things through outreach, through volunteering. I think those are the rewards, because those rewards pay forward, they pay dividends. And they also encourage other people to say, I never thought about like this, like I can actually have an impact. And so I think just the field of marine science is inherent with challenges, but if you overcome those challenges, the rewards are worth it.
Yeah, thank you for sharing, Colin. I definitely agree. Yeah, someone's giving you some extra claps. Yeah, that our field is definitely just, there's challenges woven within. And I'll have to agree with both of you that for me, that is definitely fieldwork was one of them. For my master's degree, I had to collect from the field and take it back to the laboratory. So in our aquarium room, so I'm trying to keep things from the ocean alive within the aquarium room. And just ensuring that everything is working correctly was, I think, my biggest challenge. Like, as Colin said, the ocean's unpredictable. So going out on a boat to Catalina Island, hoping that the boat is actually working, that Catalina Island hasn't lost all of its water piping and everything's broken, that the conditions are safe enough for me to be outside in the water, to ensure that I have somebody who can go to Catalina and take a full day to go to this island with me to collect algae, to fight traffic at 4:30 a.m. to 7:30 a.m. to LA, and then traffic back that night. Take organisms, pick those up, hopefully they're still alive, and then bring everything back to the aquarium room and put everything in and hope that everything is fine and that the aquarium room doesn't break down and the organisms survive. So definitely there's just all these different moving pieces when you're working in field ecology, especially if you're gonna bring the lab work into that as well. As far as, I guess, personally, I have found it very challenging to find kind of my space and where I belong in a way. And I really, I guess for me, it's been taking up space and being allowed to take up space. And then when I'm trying to build my confidence in science, being rewarded for that. Some people are rewarded for that. But as a Black woman in marine science, I was never rewarded really for speaking up or for talking. I was often cut off very, very frequently. So it was hard to really get my ideas out there and then have enough time and space to flesh them out. And although I'm a light-skinned Black woman in marine science, colorism and texturism is definitely another conversation that needs to be had within the community. But it was difficult navigating white spaces, and especially as a first-generation student, figuring out how college works, trying to access that information. And for me, it was definitely like a long process. I spent 7 years in undergrad because I just didn't know what I was doing for the longest time. As far as rewards, I would say keeping on this idea of access and having space, that when I'm able to provide that for another student who has a similar background as me, who's either URM or a BIPOC, when I can provide that knowledge to them and kind of help them the way that I didn't really get help initially, I think that's very rewarding, because as we mentioned before, it's really important to pave the way for the next generation of scientists, whether that be small children, maybe high school students or early college scientists. It's really important that we reach out and provide for them and help. And I think as far as the hard science goes, when I get I guess an experiment and it works and the data looks fine, that's super rewarding. And I was like, wow, the stars were aligned and everything happened. And another, I would say, reward is meeting people who just you have similar interests. When Katherine and I met a few weeks ago, like I was supposed to mentor her, and we just like chatted about algae and geeked out for like an hour. So like when you find your people, it's definitely rewarding, and it makes you feel like you belong, and that's really important.
I agree with what everyone has said so far and can relate to a lot of the sentiments. So I'll try to find something else that, in addition to what everyone else said about like a challenge, I feel sometimes R doesn't work. I don't know, like not just tricky, but sometimes just having difficulty like with your programming, and it's just like, what is going on? And you know, there's a story there. You're just trying to figure things out. So those days sometimes can be super challenging and discouraging, but it's like, okay, I'll just take my time and I'll figure it out. And like, it's true, you do, I think Colin said, you learn a lot through adversity, so that like the best way to learn probably is to struggle first. And I found that that's been true with myself in my own journey. So that's like, as far as science goes, a challenge. And I think like I said, I share similar sentiments just about like finding your space and understanding like kind of where you fit in this world of marine science. But I think that the longer I experience my journey, the more I feel like I'm starting to figure out where I fit in. And so a reward for me is like when you find your story, you're able to articulate it and share it. So like being able to present whatever you found, I think that's so rewarding, and actually getting it down on paper. So like I haven't yet submitted a manuscript, but just it's been rewarding to see one develop over the year and know like, okay, this is going to be a final project, and all the things I've worked on, someone's going to be able to read about it and learn about it. And also just like with students, being able to talk to them about my research and like put little nuggets in their ear about different RUs or things that might be available for them. I find that rewarding as well. So.
Yeah, thanks, Shalanda. I echo all the comments that my younger colleagues here have made. And don't let the baby face fool you. I've been in the game for a bit, so I am probably a bit older than those of you here on the panel with me. But I also want to say that I do view you guys as colleagues, right? You're all scientists in your own right, despite the fact that you're at the educational stage in your careers. But you'll get there eventually. And so I think, everything that you guys have been saying so far is amazingly insightful and important. I know, for me, that there was a struggle feeling a part of marine science, often walking into spaces where I'm the only person that looks like me. And I do work on research vessels for months at a time in the open ocean. And there are times, where I've walked into vessels that weren't as welcoming as they could be, into spaces that I didn't feel that my voice was recognized. And so all of those, I think, like you guys, were disheartening and hard to really deal with. But I persisted primarily because I had really great mentors and colleagues that continued to support me and push me to go further. I would also say, like, for me, something that's not personal, but more of a technical frustration or challenge is that the areas that I work with are expensive to get to, and they take a lot of data to really understand the patterns that are affecting the organism that we look at. So we have to go back out and do things in the same way repeatedly over time to get a really small understanding of what's happening in those ecosystems. And like, you mess up from time to time out there. And sometimes that means that like a $10,000 piece of equipment gets lost in the middle of the ocean. And that can really mess up both your day and your research for a long period of time. So, Colin's exactly right. And Shalanda's exactly right. Learning how to deal with that adversity and how to figure out ways to navigate through that is one of the things that you learn, and what makes great scientists great. I don't think I've gotten to the point where I can consider myself a great scientist yet, but hopefully one day. And navigating through the adversity is one way to get there. And also, the reward in that is that when you figure that thing out, you figure out that knowledge about that ecosystem, or you know, why those juvenile fish are being sent the way they're being sent, or how, you know, ocean currents affect that. For me, it's a really great feeling to know that I'm like the only person in the world that figured that thing out at that moment, right? And the papers that I write are justification that, like, I figured that out before anybody else. And it's a lasting, you know, effect of the work that we do in science that you get to say that. I mean, how many people can say that you made a contribution to something that no one else could have done? You know?
Yeah, so this is amazing. I'm like Carlee, when you were sharing interesting facts, I was, you know, in awe. And of course, again, with this round of questions, I share a lot of the sentiments that all of you spoke about. And I'm always so happy when we do these panels and we get Black folks on here to talk about their work, and everyone mentions education and outreach as something that is a priority to them, because that is what my career and my life is about, right? I'm devoted to education and outreach, especially for our people, especially to reach back and make sure that, you know, Black kids can see us, all of you, and look up to us as examples, and maybe learn about something that they hadn't seen before, and think about life in a new way, and think about possibilities that are endless, right? And know that you can do anything, right? Like I didn't see anyone that looked like me before. I didn't see anyone like this panel when I was growing up and chose to go into marine science. And so it is always, I talk about this every single time we do this, but it always means so much to me that we are able to, you know, come together and to uplift one another, but also to reach back. And so I'm really happy to hear you all say that I know I put so many reactions on my screen. I'm going to keep doing it because it's the only way that I can, you know, chime in without being like, yes, like I don't want to cut y'all off. So I'm just going to keep using reactions. So again, thank you so much. We're going to move on to the next question. Okay, y'all. This one is, what is a cool technique or tool that you use in the field? A cool technique or tool that you use in the field?
Well, I don't have a lot of cool techniques. I set things out, I leave them, I pick them back up. But I think something that has been really great, and I've kind of had to be a little innovative, is the loss of Tuffy pads. I don't know if anyone does any kind of settlement research here. Okay, so we have these Tuffy pads, which are like little Brillo pads, but not, and they went out of business. Every recruitment person who does little things with invertebrates has used these before. I know people in the audience know what I'm talking about. They went out of business. I'm so sure that there are like 3 labs that are just hoarding them all. And so I've had to come up with new ways to do my recruitment. And my advisor started using furnace filter pads. So the things that you put like in your air conditioning systems to filter things out. And it's been really amazing to see kind of how we're able to take things that are like everyday things that we don't really think about using, and use them and apply them to science. Another cool technique that I think is pretty interesting is the use of modeling in ecology right now. I personally am doing an integral projection model to try to figure out just how mussels are, since they are declining right now, how we're able to kind of model and project what their populations are going to look like in the future, given certain variables. And I think that modeling and like being interdisciplinary is a really important tool that we all scientists need to use to be able to solve some of our global issues.
Wow. Modeling is interesting indeed. I can't talk much about modeling. Dr. Hastes actually knows more about that than than I do. But something innovative. So, you know, my background is more in like coral ecology, but for this PhD, I've kind of stepped into a new realm studying like coral microbiology. And something that I find is really interesting as I'm starting to learn about the techniques that are being used, and the techniques that are being used in our lab, in this lab, is, just how far technology has come in our ability to sequence genetic DNA, right? Like, I remember reading how back in the 2000s, it was our first attempt to sequence the entire human genome. And, there's 6 billion base pairs, right? And so it took several years and warehouses full of machines sequencing and processing this data. Now we can do the same efforts for a substantial less amount of money in a couple of weeks. And so that is truly innovative, because these types of technologies that are usually applied to things that are directly to our own lives, so like human, diseases and research and stuff like that. But now that this has become more readily available, it's become more effective, it's more cheap, we can apply this to the marine ecosystems. And then we can really get at understanding some kind of, baseline understandings of how corals respond to their environment. And like what Katherine was saying earlier, it helps us infer how these ecosystems are going to respond when climate shifts or things happen. And so that's really what I'm really excited about. And I'm still learning how we actually do that. So it sounds cool on paper right now. Come back to me in a couple of years and I'll be able to explain it all to you. But yeah, that's kind of what I think is cool.
So for me, in my dissertation work, I haven't quite got to field work for my own project just yet, still in the lit review process of things. But within my lab with Dr. Jennifer Smith, something that our lab frequently uses, and I've been a part of helping in the intertidal zone recently, is using imaging techniques. So we use something called structure from motion within the lab. And this essentially is estimating 3D structure from two-dimensional pictures that we're taking. So within the intertidal zone, we have a project called TIDES, which is the Tribal Intertidal Digital Ecological Surveys project. And so we're essentially taking this camera and taking thousands of pictures throughout the intertidal zone within a large plot, and then putting them into a computer program and structuring and stitching all of those pictures together to make essentially this like 3D map of the intertidal zone. This is also applied to coral reefs as well. So I think it's really interesting that we can collect all of this data in a short period of time and then bring that back to the lab and spend months analyzing it. And that's really valuable, say, if you're going to work in the tropics, you only need to take 1 or 2 trips out there, collect the data within a week or so, bring it back to the lab, and then you can have a ton of undergraduates that can help out with this research. So it provides access for our undergrads to work on research, and then also it's great for grad students because then they don't have to worry so much about travel. And it's been really great for the intertidal zone because our really great tides only happen really twice a month, every 2 weeks. And the really, really good low tides might be at an even longer interval. So it's great that we can go out for just maybe 3 to 5 hours, take all the data that we need and bring it back to the lab. And yeah, so I'm really hoping that I get to use this innovative technology in the near future, maybe if I'll do some intertidal work myself and get to see where that can take research and take science.
Oh, nice to hear about that again, Danielle. I remember some folks talking about that that imaging technology that you all had at Scripps. So that's really cool to hear about. Similarly to Katherine, I kind of just take my things out into the field. I leave it there and I come back. So there's not too much technology going on, but there are some techniques that I use that I think are cool. Like, for instance, if I want to understand how much Podina has calcified or not calcified, depending on the treatment that I give it, I kind of decalcify it. So that's the process of just like essentially pouring diluted acid on it to like get all the calcium out of like the carbonate out of the algae, so that I can see like what does that change in weight. So I think that's something that's really cool. The most technological thing that I've used recently are hobo loggers. So they're just like these little data pieces that you put in the water, and they can help you gather information such as like temperature and like light information. And so that's been really fun to use because otherwise, you know, there's not too much gadgets I would use. So I thought that was really fun to be able to help characterize my site. So.
Yeah, I would say for me, I think the most interesting piece of technical equipment I get to use are the research vessels that I get to do my research on. And like when I tell my friends about the work that I do out there, they're like, oh, so you're going back out on the boat. And I'm like, I'm going out on a ship, okay? These are vessels that range from 40 feet up to 80 or 90 feet, and they can host upwards of 40 or 50 people to 80 people, all living on a vessel out in the middle of the ocean together, for weeks at a time. And these vessels can do a range of amazing things. In the United States, we have two main systems of research vessels, right? There's the US academic fleet, which is managed by NSF and the University National Oceanographic Laboratory System. I think I got that right. And those are the more academic research vessels, which include, icebreaker ships that go up and study in the Arctic. I do a lot of work with the National Oceanic and Atmospheric Administration, so NOAA, and they have fisheries vessels that operate in all of the coastal and open ocean waters of the US, studying fisheries as mandated by Congress, and so I get to do a lot of research with them. And those vessels for us tow nets, and I'm not saying like a fisherman's net, but we tow like highly specified nets that allow us to collect information on the ocean and collect organisms that we can study while we're out there. These include nets that can sample discrete ranges by depth in the ocean, right? So, one called the Mach nest is I'm never going to remember what that acronym stands for. But the cool thing about this net is that it has these sliding gates that shut depending upon the depth of water it's in. So you can set it to program and sample or be towed behind the ship, between 50 meters and 40 meters, and then close this gate shut. So I've collected larval fish only for that range in the water column. And you do all of this from like a control room in the ship while this thing's being towed behind the boat. So, for me, I think the research vessels, that's part of the reason why I got into this and why I stay in it, is the opportunity to be out in the middle of the ocean with like 30 or 40 other people and see nothing but water, stars around you, and try and understand something about this blue marble that we live on,
Awesome. I loved learning about all of you guys, different techniques that you use. You guys are just awesome. All right, so the last question we're going to give you guys is, what is something that you're interested in studying that falls outside of your area of research?
Ooh, outside of my area of research, I think I'm really interested in, again, like going back to that outreach, and I'm currently a teacher, so I'm really interested in like the sociology behind how people interact with the ocean and vice versa, and really looking at those. I mean, I guess this isn't really outside of my research, thinking about like ecosystem services. But I am really interested in like who gets those services, who has access to those services, and kind of what that interaction looks like, and how we can then shift, what we study, how we study it, and what kind of community interaction we have to be able to really make an equitable coastline.
Yeah, man, again, to echo Katherine again, something that I feel something that I'm very interested in learning how to do that's outside of like marine science or just STEM in general is effective storytelling. It's communicating. I have been really lucky throughout my experiences to take a lot of like underwater GoPro footage and it's like a lot of things, and I'm trying to learn how to encourage people to find interest and get excited about coral reefs, encourage other people, especially minorities and people who have been marginalized out of this field, to say like, this is something that you can do, this is applicable to you, but then also to try to make a connection that studying the ocean can really benefit communities around the world, but then also the ecosystem as a whole. And so trying to develop stories and trying to communicate with people to emphasize the importance and use fun and innovative ways. Like right now, we are in a technological revolution, right? Like a lot of our experiences are online, and a lot of our experiences, what we're learning from is in this kind of platform. And so trying to be more efficient at trying to educate people, and because we need it. We need people to step up and to join the career in marine science, especially people of color. It's important to have diversity, right? That's the only way we're going to be able to accomplish the goals that we have. And so how to communicate to a broad audience to encourage them is kind of what I'm interested in doing.
Awesome. Thank you for sharing, Colin and Katherine. I also agree that community outreach has always been an interest, but so I will avoid repeating the knowledge that was already shared. But something that I'm really are becoming interested in is genetics. So I've actually feared genetics for most of my undergrad and master's degree and actually avoided it at all costs. Although I had some really cool friends that were geneticists. And I just didn't want to get into it. But within just the world of seaweed and classification, seaweeds are always being renamed. And it's kind of irritating as someone in ecology. Right before my thesis, my master's thesis, there's a species that's named Icenia arborea, which is actually in my wall right here. And so it's this very beautiful species. And like right before my thesis was supposed to give my presentation, it was changed to Eclonia arborea. And so my PI is like, oh, well, you have to change that when you're presenting and defending your thesis. And I was like, oh, all right. So I spent an entire day going through all of my R script and changing all of the figures name. And it was kind of irritating, but I understand the importance of it. And so for non-native seaweed species, there's still some questions about, well, we have a seaweed species. It might be a non-native, but it looks very similar to the native species. So really genetics is the way to answer those questions. And that's something I'm kind of hoping to dip my toes into over the next few years.
Thank you all for sharing. So something that I think is interesting outside of my area of research is specifically with macroalgae or just algae in general is understanding how to use them for different purposes, such as like medicine and like biofuels. So I find that to be very interesting. So I'm interested in learning more about that and how people kind of apply the things that they learn from that field and help to other people like in the world and in that broader scale.
So yeah, thanks, Shalanda. I think I'm gonna have two. I'm just gonna sneak in two there. But so first, I'm gonna echo Colin's comments. So some of the most interesting work I'd say that I'm working on right now is looking at structural practices or figuring out evidence-based strategies that we can use to affect structural change towards justice, equity, inclusion, and diversity in the geosciences. Which I'm a part of because oceanography is considered a geoscience. And so, part of what that is working with sociologists and anthropologists to understand the practices that they use to model and study, human behavior. I never was trained in that. I don't understand that. I'm an oceanographer. And so what it requires to do that work is to create transdisciplinary teams that can work together. And I have to say, I'm learning just really interesting and really cool techniques on a few of the projects that I'm working in that's related to that work. The second one is, again, related to transdisciplinary things. So I have another project, which is a national research traineeship. So it's training our students here at the University of the Virgin Islands to look at the food, energy, water system. So it means rather than looking at things as silos, right? Food production, energy usage and production, water desalination, water use, water movement. Don't look at them as separate systems where we have environmental engineers and biologists study them separately. These have to be looked at as combined systems where everybody is working on the problems that affect them together because they're all related. And so again, that means me stepping outside of my comfort zone, which is oceanography, to learn new tools and techniques from other researchers who are way more experienced than I am. And I think, both of those have taught me a wealth of knowledge and has really inspired me to move my research into different pathways, but also to find ways to connect what I do now with those things, both justice, equity, diversity, and inclusion, but also within food, energy, water systems.
Okay, thank you everyone so much for your time today. Again, it is a pleasure to host this panel, and thank you to the organizers of hashtag Black and Marine Science Week for reaching out to National Aquarium for us to host this. We are always happy to do these sorts of events, and we are happy to support the efforts. And thank you to all of our panelists for sharing your experiences, for being open, for talking about your interests and the things that are really important to you. That means a lot, and I know that your story will resonate with others. So really happy to have all of you here, and I'm really happy to meet y'all because I haven't met any of you yet. So I'm really happy that I met y'all now during this panel. And I also want to say thank you to my co-moderator, Carlee. Thank you, Carlee. I appreciate you. Yes.
No
Round of
problem.
applause. Yes.
Thank you for letting us host this with
Yeah,
Black and Marine Science Week.
Girl, of course, of course. And so thank you to the audience for watching. Thank you for supporting hashtag Black and Marine Science Week. And we want to, of course, plug hashtag Black and Marine Science Week. So Carlee, do you want to shout out our socials?
Yes. So the hashtag for the whole week is going to be hashtag Black in Marine Science. And then our Instagram is at Black in Marine Science. And our Twitter handle is the same, at Black in Marine Science. So go check those out. We have a schedule that just lists, everything that's happening that week. So yeah, check it out.
Thanks, Carlee. And again, thank you to all of our panelists, and thanks to the audience. Have a good one. Bye, everyone.